Unit 5 covers Chapter 6, which is, well, one of our most technical and specific chapters. We'll try to walk through this step by step and make it as understandable as we can. We wanted to step back and start with a little review of photosynthesis, primarily because the focus of this chapter, cellular respiration, really feels like the opposite direction of this reaction. Look at photosynthesis. In it, we take carbon dioxide, add water, and produce glucose and oxygen utilizing solar energy. Realize that plants and other photosynthetic organisms, called primary producers, do this by using a series of enzymes and pigments to absorb that solar energy. The reaction for cellular respiration really looks like the reverse of photosynthesis. Glucose plus oxygen is converted into carbon dioxide, water, and ATP. The only difference is instead of solar energy driving the reaction, we produce or convert into cellular ATP energy. This reaction averages about 36 to 38 ATP per glucose molecule. That conversion is guided by the first and second laws of thermodynamics. Recall the second law. Every energy conversion is not as complete as we'd like. If so, for every glucose molecule, perhaps we could get 100 ATP, but instead we only get about 36 to 38. Think of that as a percentage, and compare that to, well, the efficiency of energy conversions and other things, like our cars, which average about 25 to 30%, or you think of it as miles per hour. Throughout this chapter, we'll be removing hydrogens and electrons, and the cellular reactions that do that are known as oxidation and reduction. Oxidation is when you lose an electron, and reduction is the opposite, when electrons are gained. We'll couple these two reactions together in what are known as redox reactions. In the diagram on the right-hand side, you can see where the reduction of the oxygen and the oxidation of the Fe iron produce a coupled reaction with iron oxides being the products. Now, the real focus of this chapter is cellular respiration, and the term respiration implies there are gases involved. Recall that this is in the cells, not in your lungs, so we're at a much more microscopic level. There are 3 steps involved in cellular respiration if we're going in the aerobic pathway. They include glycolysis, citric acid cycle, and oxidative phosphorylation. In each of these, we'll be moving substrates and products through the cell in an attempt to convert the food we eat into ATP energy. Before we dive into those steps, I want to review electron shuttlers. These are molecules, most of them made from the vitamins we consume, that in essence are carrying or binding and then moving and delivering both hydrogen ions and electrons. These include NAD, FADH, and you'll come across names such as electron carriers and electron transport chains. All of these are going to help in producing ATP. ATP can be produced in several ways. The two that we'll discuss connected to this process are substrate level phosphorylation and oxidative phosphorylation. In substrate level, an actual phosphate comes from the molecule and is attached directly to the ATP. In oxidative phosphorylation, free phosphates are then bound with energy that is coupled from other sources. Let's dive into the first step of aerobic respiration. That's glycolysis. This is a universal process. It happens in all cells of all types, prokaryotic and eukaryotic. In the word glycolysis, it literally means the splitting of glucose. So glucose is split in half to make two pyruvate molecules, also known as pyruvic acid. This occurs in the cytoplasm in a 9-step process that involves both enzymes and energy input. That energy is in the form of two ATP molecules. This is known as activation energy. It's the amount of energy to really be inputted and basically phosphorylate the actual glucose molecule in order for the production of that pyruvate. We will also get hydrogens and their electrons removed in the splitting of the glucose, and those are picked up by Now, I'm going to give you both a simplified and a more in-depth view of glycolysis, but please note, we only expect you to be able to learn the simplified since we don't have enough time to really spend on the full process. So, as a short overview, glucose in this diagram is being enzymatically broken down, and I've split it into two sections. A mid-intermediate product that is shown as a 2 3-carbon molecule that is phosphorylated using the 2-ATP activation energy. That intermediate product is further broken down with hydrogens removed, see the NADH being produced on the right-hand side, and ultimately 4 ATP molecules are produced, and 2 pyruvates. So glucose is converted into 2-pyruvate, 2 ATP molecules are put into the system, 4 ATP molecules are produced, and 2 NADH molecules are also produced. Notice we make 4 ATP molecules, but we needed 2 as activation energy. We have to repay these. It's kind of a loan. So our net gain of ATP is only 2. And remember, this takes place in the cytoplasm. This more in-depth diagram of glycolysis allows you to step by step see the conversion of glucose to pyruvate. You'll see the input of the ATP, which allows for that substrate level phosphorylation. I would never expect you to understand each and every step and be able to reproduce that on a test. That's just beyond the scope of our class. But for those enthusiasts that want to see it step by step, here you go. Regardless of the detail, we end with 2 pyruvate molecules from glycolysis. Those pyruvates must be modified or groomed if they're to continue down the aerobic pathway. Otherwise, they're destined for the anaerobic pathway. In the aerobic pathway, the grooming involves the removal of carbons and oxygens and hydrogens. The carbons and oxygens are removed and modified into carbon dioxide. The hydrogens removed are separated from their electrons and both are carried or picked up by one of those electron shuttlers, NADH. In place of those carbons, we attach a coenzyme A. Coenzyme A is a vitamin B derivative. When we do this, we alter pyruvate into a new molecule called acetyl-CoA. Because we make 2 pyruvates, we then make 2 acetyl-CoAs. If acetyl-CoA is produced, we then can move to the next of our major steps called citric acid cycle. This takes place inside of the mitochondrial matrix. Remember, that's the fluid. So we have to physically move from glycolysis in the cytoplasm to across the double membranes and into the mitochondria. All the remaining carbons that are in those acetyl-CoA molecules are going to be disassembled. So we're going to end with lots of carbon dioxide. This is the predominant area that carbon dioxide is produced. Notice from the diagram at the bottom, we're also going to produce 2 more ATPs, and all the remaining hydrogens will be picked up by NADH and FADH2. In a simplified diagram, I want to show you that the acetyl-CoA is entering into a cyclic enzymatic process. That means the molecules that are picking up and binding acetyl-CoA will go through a series of changes. They will kick out carbon dioxide, hydrogens and electrons, and ATP, but those molecules themselves aren't altered, and they step by step go through cycles that then come back to receive a new acetyl-CoA. There are many enzymes involved in this pathway, but ultimately our products are carbon dioxide, ATP, and those electron shuttlers, NADH and FADH2. In this detailed diagram of the citric acid cycle, you can follow through the carbons as they are passed through the cyclic pathway of the citric acid cycle. Remember, this takes place in the matrix or fluid inside the mitochondria. We are removing hydrogens and placing them on carriers, NADH and FADH2. We also produce carbon dioxide and ATP. The majority of carbon dioxide our bodies produce is in this cycle. The last step of aerobic respiration is oxidative phosphorylation. This process actually has two steps involved within it. They are the electron transport chain and chemiosmosis. In the electron transport chain, all of those electron shuttlers bring those hydrogens and electrons to the folded up inner membranes inside the mitochondria. Remember, these are called cristae. They will unload or oxidize, releasing the hydrogen ions and their electrons to the proteins embedded in the membranes. Those membranes then will use the energy from being sort of electrified or excited from the receiving of those electrons, and this energy will allow them to pump hydrogens across the membrane and create a gradient of high concentration. In this diagram, follow from the lower left corner a NADH molecule arriving and donating both the hydrogen ion and the electron to the first protein in the electron transport chain. The electrons are energizing the proteins. Really, this means a conformational shape change. Ultimately, what's happening is the hydrogen ion is pumped across the membrane to the upper level where hydrogen ions are going to be in higher concentration. So we're going against a gradient. We're creating on the other side of the membrane an area of hypertonicity. So this is an active transport mechanism. Remember, this is called a pumping process, and it takes energy. Instead of ATP, though, we're using electrons to energize these proteins. FADH2 can also come in and drop off hydrogens, so all electron carriers or shuttlers are arriving and donating their hydrogen ions and electrons in this process. This is the electron transport chain, and the end products are really hydrogen ions in high concentration. Now that all the hydrogen ions are in high concentration on one side of the membrane, very specific protein channels can be opened. These channels, known as ATP synthase, allow for hydrogens to flow down their gradient. So this is a passive process. As they flow down, though, these special protein channels couple a process of phosphorylating ADP and produce ATP. This process allows for the hydrogens to come back through the membrane to be rejoined with their electrons and finally joined with oxygen to make water. In this diagram, follow the little red hydrogen ions in higher concentration in the upper membranes as they flow through the ATP synthase. Notice the hydrogens move through, and in doing so, ATP is produced. The hydrogens are rejoined with their electrons and oxygen to make water. This is where the majority of the oxygen we breathe in is utilized. Oxygen is the final acceptor of both the electrons and the hydrogen ions. Remember that the pyruvate, also known as pyruvic acid, must be groomed in order to go down through the aerobic pathway. If it cannot, then it's destined for the anaerobic pathway. So we still have glycolysis taking place, but it's the fate of that pyruvate. In the diagram here, you see how in animal cells, like our own, pyruvate is converted to lactate, or known as lactic acid. All is done is we put back on the hydrogens that were removed in glycolysis. Please note, if this was a plant, a yeast, or a microbe, the product would be slightly different. Their enzymes don't end in lactic acid, but rather they produce ethanol, also known as ethyl alcohol, and a carbon dioxide. So the difference is really in the enzymes. To compare and contrast aerobic and anaerobic, remember that both processes start with glycolysis, and both are breaking down glucose. But what's different is they're at that pyruvate stage. Aerobic produces somewhere between 36 to 38 ATP. We do so with predominantly most ATP being produced in oxidative phosphorylation. Anaerobic respiration only gets 2 ATP total, and those are from glycolysis. No extra ATP is produced in the pyruvate conversion to lactic acid or ethanol. The entire process we've discussed has always started with glucose, but that's just because it was first discovered using glucose, and glucose is our most widely used sugar. It's the most common sugar produced through photosynthesis. Any monosaccharide can really go through this pathway. It depends on the organism and what enzymes you have. But what if you're not using carbs or sugars? You could insert fats or proteins in various locations along the pathway. Note that proteins are broken down into their peptides or amino acids, and in doing so, we must remove the amine groups. That process is called deamination, and those amine groups produce ammonia, which is highly toxic. So metabolizing proteins produces some pretty toxic products. Amino acids can be inserted, depending on their size, into either the pyruvate stage, the acetyl stage, or directly into citric acid cycle. We generally, in sequence, tap into our carbohydrates first as energy, and the proteins as second. Why not the fats? Well, remember, they're hydrophobic. It's difficult to get them to go through these processes since everything is in the cytoplasm and mitochondria in a water-based environment. In order to use fats to metabolize, we have to, 1, make them more water-soluble, and we have to break them down into glycerol and fatty acids in such a way that they can be inserted somewhere in between glycolysis and acetyl-CoA. It's just difficult time and energy to make those fats go in through these processes. That's why fats keep getting stored and are less, tapped as energy sources.